thank you for the kind invitation to participate in this webinar. It's my pleasure to share some thoughts on a big picture aspect of complex generics, and that I am framing it as conquering complexity in complex generics. My goal is to share with you my experience and to frame the key challenges that exist in a way that will help Dr. Tissot's technical aspects to be placed in the overall context of complex generics as we see in the real world today. The big challenge really is our ability to appreciate complexity and to be system oriented. I think that is something that needs attention, and I wish to call your attention to that in the context of an evidence architecture, which is key to assurance of therapeutic equivalence within the scientific community, within the regulatory community, but also provide the assurance patients and providers need in the real world. So my talk really is divided into background, challenges, questions, and answers. And the background challenge that I see is we have to seek equivalence in an inequality sort of a mindset as well as economics. And the challenges that I see are in our mind, methodologies, and matter. And the question of sameness, of expectations, and of materials in the context of the system that we currently have for development, manufacturing, and for insurance of generics. And that's I'm talking about health insurance. And the key then is care is research, knowledge is power, wisdom is practice. That's really the key. Equivalence in inequality is a paradox that has existed within our sector for a long period of time since the establishment of Hatch-Waxman and the ensuing generic drug scandal and the continued challenges that we have in assuring equivalence to the satisfaction of all the patients, all the providers out there. So in that sense, I think it is important to remember by definition, if you carefully read the Orange Book definition of therapeutic equivalence, it is an expectation. And the words sameness and similarity and aspects of equivalence that we talk about in the technical community or the scientific community have different meanings in the real world. And in the 21st century, particularly under the 21st Century Cures Act, we are facing an opportunity, but also a challenge of real world evidence. And I think I want you to place this discussion in the next 10 years of real world evidence, value-based pricing, pay for performance, outcome-based reimbursement that are really shaping the economic dynamics of our sector. So the challenges are 3 M's: mind, methodology, and matter. In the context of sameness, system, and substitution, and automatic substitution, and the challenges that you see between complex generics and biosimilars in Europe, US, and elsewhere, I think the yearning to resolve the tension between sameness and similarity really is at the core of the challenge. So the questions really are sameness of expectations and materials. How do we achieve that? And system for development, manufacturing, and insurance. How do we accommodate, how do we manage these systems that are ecological systems that we live and work in, and we have to work towards improving the systems from within. So the key outcome we desire is substitution interchangeability based on hard scientific evidence. And that's really the yearning and the goal we all have, is to provide sound scientific evidence for all of our claims. To help appreciate the challenge from a big picture perspective, let me share with you a few milestones in my career. I think, and which are directly related to the complex product manufacturing, development, manufacturing, and approval. So I have selected to put this, provide a context with four of these. Apotex launches first generic version of Merck's Nasonex in 2016. FDA approved Sandoz Omnitrope as follow-on protein in 2006. Sandoz leads the way with its first generic version of gold standard antithrombotic Levonox, enoxaparin, and I think Dr. Tissot will build on this analytical challenges on this in her talk. And something which is happening, unfolding as we speak, an evidence-based approach to tobacco conundrum, I think also an example which I felt is appropriate to think about, where I think we are seeking to demonstrate sameness, equivalence, or similarity to harm reduction as it occurs in the context of smoking cessation. And so that's the reference out there is not a product. The reference out there is prior knowledge. And so I think in the pharmaceutical sector also, in fact, I'm sort of working on a project right now where I don't have a reference product. And how do I guide this company to develop equivalence, sameness, evidence architecture when you don't have a reference product? So I think, so I'm going to use these examples as a way to frame put the two questions I posed earlier in the proper context, and to highlight that going from the system that we have currently, whether it's a corporate management system or a regulatory system or a healthcare system or the insurance system, how do we work within the system to appreciate complexity and to build an evidence architecture that satisfies the outcome we all strive for, that is scientific evidence-based decisions and claims. And clearly, I think we want to move in the direction challenges leads to good evidence to opportunity. And but the challenge what we have is the feedback mechanism. There are too many failure rate. The failure rate tends to be high. And how do we avoid the negative failures that we see in the system? So let me highlight an example, more recent example of the generic Nasonex, which was approved in 2016. What is so noteworthy about this approval? And I'm using FDA's own words to do that from this publication that they presented. FDA embraces emerging technology for bioequivalence evaluation of locally acting nasal sprays in lieu of the clinical endpoint study, which was deemed unacceptable because of API related differences in particle size. And FDA used in lieu of clinical evidence a Raman directed spectroscopy method for particle size and shape. This was first in the history of Office of Generic Drug. Their words. The challenge with complex generics is we have to bring pieces of evidence from pharmaceutical equivalence to bioequivalence, in this case, equivalent in vitro performance, equivalent PK studies, equivalent local delivery, in a way to bring totality of evidence. And totality of evidence, integrating different pieces of evidence from in vitro, clinical, and so forth, we struggle in that mode. So here, I think two messages that come about, and I'll elaborate on that. Pharmaceutical equivalence is the key. Pharmaceutical equivalence defines, justifies bioequivalence. And therapeutic equivalence is not only an expectation, it is a system of different parts. And I think we often forget that. And the key lessons that I take away from this example is good methodological evidence cannot be complex, and good material evidence should be self-evident. And that's where we struggle with the current system of pharmaceutical equivalence. The definition of pharmaceutical equivalence when it comes to dosage forms and other aspects is very vague and old. It's not up to date. And complex is something where the cause and effect relationships are known after the fact or in hindsight. And that's really where I'm going to take my discussion forward. The other example that I want to share with you is one of the reasons I left FDA was to take on the challenge of this complexity, was to open the door for biosimilars, and the key challenges here were uh, inability to bring together different pieces of evidence of comparability and analytical characterization to not on the technical aspect but to open the door for political consensus on moving forward with complex product equivalence, sameness, and similarity consideration. So this became a very important milestone in that, and FDA's response to the citizen petition is a landmark document. So in this case, I think this was where I fully realized that blocking citizen petitions can become an opportunity for some companies, and that pharmaceutical equivalence in and of itself is complex, but this complexity is not being appreciated. The true application of that understanding is sort of evident in the approval of the first generic enoxaparin, Sandoz Momenta, and FDA itself has written a paper on that, scientific considerations in the review and approval of generic enoxaparin. It's a very valuable paper, and I really urge you to get that paper and read that. And Dr. Tissot will build on that. But the key here is we need new prior knowledge, research in a generic company, investment in research. And here, I believe we were blessed in the sense we had Momenta and MIT's prior research, prior knowledge to build this application on, and that was key to success. New prior knowledge is key, and I will build on that. One thing which we have to appreciate is we in the world of complexity, we have to reduce complexity by going way beyond typical QC tests, way beyond compendial testing. So in that sense, the sameness equivalence criteria, particularly the in vitro one, is based on more exquisite orthogonal set of analytics that need to come together. And the challenge is the more you look, the more precise and more accurate and more sensitive analytical methods you bring to bear on the problem, the more differences you see. So that's the paradox one has to resolve. And in that paradox, one of the key aspects early in the learning of enoxaparin development was, let's not overlook something that we always do. 3 lots of RLD is enough to, no, we need to understand the variability in the API of RLD and the API of the product, and we have to get the right number of lots to understand that source of variability, which is often overlooked. But also we need to be aware of there will be continued scientific scrutiny, because in 5 years from now, there'll be new analytical methods, and people will bring new analytical methods to show there is difference. The products are not the same as is occurring now. And so you need to have a defensible statistical analysis and acceptance criteria that will withstand the test of time when new analytics come in and show differences between products. What differences matter, what differences don't matter is a key. And that became a leveraging opportunity for at Sandoz to bring a systems approach, a research analytical characterization investment approach to be able to show variance in the RLD that does not matter. And I won't get into the detail here, but I think if you want, I have given a reference, you can get more detail. Key to success here is to not be confused about complexity and legacy blindfold in the generic system. And I think that's my key message to you is take a few moments. It doesn't take too much time. Step back and look at, re-examine your assumptions you're working under. That's it's simple as that. Is to re-examine the assumption and question, ask why 5 times. I mean, we have a beautiful tool of 5 whys for root cause investigations when we get a out of specification result. That same mentality is you have to start with that. Ask 5 whys before you start your development project. And I think that's really the key message that I wish you to take back. This is very important because we often don't do this. We work with assumptions. We work, which I call blind spots. Unverified assumptions are blind spots or blindfolds. And I think this is this is evident widely within the sector. So based on FDA's own statistics and my own experience at FDA, our generic system functions at 40% efficiency. For 10% of brand products which have expired patents, we don't even have ANDA applications. Of the 50% of ANDAs approved, uh, very few are marketed or not marketed at all, or marketed sporadically, or marketed after delays. And of the 40% that are marketed, some are, a majority are in compliance issues. So I think this is a fundamental aspect which I uh, request your attention on, because the solutions are right, hidden in plain sight. It is our assumptions that we don't. Put this in a proper context, let me paint a big picture. And this is a big picture painted based on the current scenario. So point number one: the US generic drug market in chaos, Indian upstarts rise. This is the Bloomberg report 2017. A few years ago, generic pill color changes may affect medication adherence. The placebo and nocebo effects have been skyrocketing in US and elsewhere now. And on the big picture side, fed up with drug companies, hospitals decide to start their own. Now, suffocating with silence prevented us from questioning the rot in the system. That's from Ranbaxy's situation. So that's the system above where we live in. So system within our regulatory system, FDA moves to increase generic competition. Continuous manufacturing has strong impact on drug quality. That's a wonderful opportunity that the public health need is there to increase competition. But if you increase competition without asking the right question, you only add to the eroding confidence people have in generic drugs. Appreciating complexity and to be systems oriented is the key solution in the management, in the mind of our professionals who engage in these activities. So here I'm simply showing that the real world evidence is based on real world data. Dr. Woodcock's paper on real world evidence and real world data and evaluating drug safety and effectiveness. And I think the implications of that for generics, follow-on proteins, biosimilars, is something that will evolve over the next few years. And the development of these products take time. So we need to be aware of these challenges is my recommendation to you. Let's not underappreciate complexity. Let's not put quality by design in the same old box. Take a few moments, take some time to reflect on our assumptions. Don't discount expectancy effects. Placebo and nocebo effects are active. Our clinical trials, we take extreme precaution of double blinding and placebo control arms to account for placebo effect. Do these effects go away? No. So in the evidence that we have today is that about 20 to 30% of the population in America have no trust in generic drugs. 70% do. But we have to have an education program, which FDA is really engaged in, and we have FDA has always engaged in the education. That's very important. But the 20 to 30% of population is a critical part of our system. We can't ignore them. So public health promotion of generic drugs and warning letters, recalls, and so forth that erode confidence clash, and we need to avoid that clash. In that sense, also, I think for companies such as Sandoz, when I joined Sandoz, we saw a major opportunity to collaborate with FDA to open the door to first generic. Some companies have that capacity and capability and leadership within their organization to be able to propose a way forward. Even though there is a blocking citizen petition, doesn't matter. If you put forward a scientific development plan that is convincing, that opens the door for collaboration with FDA. You don't have to wait for FDA guidance to do so. Clearly, FDA guidance will come later, but with the number of meetings openness that FDA has offered, not only in biosimilars, but now in complex generics, engaging with FDA, putting forward your proposals, then becomes a way to accelerate overcoming citizen petitions, overcoming the blockages, and actually to be more competitive. Next. Every system is contained in a larger system. I started with the definition of complexity as when cause and effect are known after the fact or known in hindsight. Let's remember four types of systems: simple, complicated, complex, and chaotic. And let's differentiate systems from disorder. In the context of cause and effect, in a simple system, cause and effect relationships are self-evident. Check the box, best practices work. Complicated, expertise needed, and you need plan, do, check, and act. This is where good practices work. Complex is where cause and effect relationships are unknown or known in hindsight. Here we have to adapt careful propensities and disposition. And in research and development, we have to adopt this careful propensity and disposition so that our development leads to a way to manufacture it in the good manufacturing system. And the product is so designed that it is self-evident to the patient in the sense they assess equivalence. Chaotic is not disorder. Chaotic means extreme sensitivity to initial conditions. And disorder is not knowing which system and how parts of the system we manage interact. I wish you would take this as a way to think about that. David Snowden and others have written on this in Harvard Business Review. It's a very useful document and contribution that I think would help you to think about framing your challenges. So in summary, a sameness of expectations and materials, systems for development of manufacturing and of insurance, which we don't control, is to understand systems, to reduce complexity, to build forward evidence in an architecture that makes sense. So wrapping up my talk, let me summarize. State of our mind matters. We need a learning space. This is a reflection of my discussion debates in the ICH Q8 step 2, where I think I had pushed hard for design space and talked about epistemological aspects and to recognize the Procrustean aspects of our human behavior. And I'm really working on how to build a learning space. That's the reason for my commitment to professional development. That's much of my work today is on professional development of from CEOs to people in the front line. That's what I'm focused on. Care is research. Invest in new prior knowledge. I think if you look at some of the work Sandoz had done when I was there, and perhaps they're still doing it, is invest in analytical characterization early and know the differences that matter and that don't matter. It's very important. Knowledge is power. With multiple complete response letters, FDA inspections, and so forth, is a challenge that I see not only in the generic, but this is a case example that I see of quality systems where immature, complex, and highly paper based, and so forth is widespread. Like evidence architecture is key. Think about design, quality by design of your evidence early before you start your development by investing in research and analytical characterization to know what matters and what doesn't matter. And ontology is to know reality of sameness or similarity in the context of patients, profiteers, professionals, and public. And I think remember, therapeutic equivalence by definition is an expectations. As a scientific community, we have our expectation, but our expectations have to match with the expectations that providers and patients have in the real world. So you have to consider aspects of that in your development planning. And epistemology is a way to question your assumptions. How do you know what we know about therapeutic equivalence and about RLD? Methodology then comes from that is how do we generate new prior knowledge on therapeutic equivalence for our test product or our generic product? And then the methods have to be suitable and capable to measure the differences that matter. Thank you for listening. It's easy to say what I said. Wisdom is in practice. Our problem is not technology. Often our problem is us. Thank you for listening.